First of all, I would like to send my warm regards to all the colleagues in Brazil, which I estimate very much, and have been participating in many of your meetings. Now, concerning the new data on Helicobacter pylori from this meeting at the AGA, the most interesting I take away from here are new recognitions in the mechanisms of immune responses that are induced, so many molecular mechanisms from the infection which can be studied very easily in humans because the stomach is accessible by endoscopy. And for instance, I was reporting here on the model of acute infection of healthy subjects with Helicobacter pylori. We induced gastritis, and all the people experienced symptoms which disappeared quickly. So we have insight now on what happens when Helicobacter is acutely infecting the human stomach. And in the process of elaborating now the immune responses, which I cannot completely anticipate, we know that there are many complex issues. The session this morning was fully dedicated to treatment, and the focus was on alternative treatments for Helicobacter pylori, since the traditional triple PPI amoxicillin clarithromycin has reduced its efficacy because of the emerging resistance. So there was a huge debate, mainly studies presented from Europe, Italy in particular, showed that there are ways of overcoming resistance by either giving 4 drugs altogether, the so-called concomitant therapy, which is as effective as the sequential therapy. And I could just say, recently in Europe we also did a very large study on quadruple bismuth therapy with a new galenic formulation, and we reach now eradication rates which are superior by 30% compared to the traditional PPI. So we have sequential therapy, we have quadruple concomitant therapy, and we have classical bismuth-based therapy in new galenic formulation, which are the new therapies that one could take away from this morning session.